So now I want to talk about the evolution of the Y chromosome, or the sequence evolution on the Y chromosome. When I say Y chromosome, that also includes the W chromosome in the ZW system. So, but we want to talk about the degeneration or the evolution of the Y chromosome. So after the differentiation from the proto X and Y, the Y chromosome, so usually we just say the Y chromosome become degenerate and also evolve rapidly. That means they accumulate mutation more rapidly than their original homologous counterparts, the X chromosome. One of the reasons for that is there's more cell cycles in male germline. So think about compare the spermatogenesis and oogenesis. I think it's true for Drosophila and mammals. There's more cell cycle during spermatogenesis before forming the final gametes. And that illustrates if every cell cycle there's DNA replication, right? Mutation rate, even given the even mutation rate for every DNA replication, if you have more replication, there's more chance the genome will govern more mutation, more new mutations. So that's one of the reasons the Y chromosome can accumulate more mutations during evolution. So there's a higher mutation rate. The X chromosome has been through the same process, but think about the X chromosome and all the other autosomes also has some time in the female. So it seems equal, like they spent some time in the female and also in the male. But Y chromosome is the only one, like they only evolve in the male. So that's why the Y chromosome have higher mutation rates than the X chromosome. Another thing is also obvious is the effective population size. Okay, in the very early of this class, we talk about effective population size because population size is one of the important factors to shape the allele frequency change during evolution. And the effective population size is different between X and Y chromosome, which is also obvious because each female has 2 X chromosomes, but each male only have 1 X chromosome. Compared to autosomes, all the autosomes, we all have like 2 pairs of autosomes in our cell. So the effective population size is different compared to the X chromosome to the autosome. But also the effective population size for the Y chromosome is much smaller than that of X chromosome or autosome. The same reason because the Y chromosome only exists in males, or I should say this Y or W chromosome only exists in the heterochromatic sex. So the population size, the effective population size is reduced for this chromosome. And reduced size will result in the reduced diversity. Okay, and there are 4 different models talk about the Y chromosome degeneration. So these models are very similar. They try to illustrate why the Y chromosome degenerate quickly after the divergence between the proto X and Y. So the first one is background selection. So background selection is the selection to eliminate deleterious alleles. And so showing in this slide, you can see the selection against deleterious mutations in the population. Then at the end will result in sort of reduced polymorphism in the population. And the second is Mullis-Richard. It means Mullis-Richard is a process which the deleterious allele occurred in the chromosome region, but they cannot, because of lack of recombination, they cannot recombine out. So the slightly deleterious alleles will accumulate in the population and over time. So the number of deleterious allele will increase in the population. So more chromosomes will have deleterious alleles associated with this chromosomal region or with this chromosome. And the third one is genetic hitchhiking. The genetic hitchhiking sometimes is a lot of people compare genetic hitchhiking and background selection side by side, but one important difference between the two is the genetic hitchhiking, the selection is on the beneficial allele showing in blue, on this showing by blue in this slice. So you can see the selection favor the mutations, then they work beneficial mutations. So then by fixing the beneficial mutation in the population become more monomorphic. So the reduced polymorphism in the nearby region is more obvious than the region in the distant region of this part. So you can see the red, the small red close to the green are fixed in the population. That's a neutral mutation. But a little bit distant from this region, you see like some polymorphism. So that's also another mechanism for reduce the polymorphism in the population. The last part is the selection against deleterious mutations. And this selection reduced against the deleterious mutations, even just reduce all the, so that the black chromosomes on this slide is showing like the chromosomes with linked beneficial mutations. So this is like possible mechanisms or models for the Y chromosome degeneration. How about the X chromosome? So we talk about the Y is largely degenerated, and there's a lot of underlying mechanisms shaping the Y chromosome evolution. And in general, X-linked genes evolve faster than compared to genes on the autosomes. And we already talked about the possible the reasons. There's population size differences, and there's a lot of study compare the evolution or the population polymorphism between the X and autosome. So here is sort of a summary from one of the review paper talk about the process of the X and Y chromosome during evolution. And I think you can just look at the figure and try to understand what was illustrated here. This is like more try to put all the possible evolution forces and the results on these slides.